All right, now that we have our formulated research idea, so that's our umbrella. Pretend that's an umbrella. Now we will come up with our specific aims that are nested under that umbrella. Oh, there it is, the umbrella again. Okay, maybe I should come up with new analogies. So the specific aims that we're writing, what they're doing are addressing different elements of our broader research question. So again, they're falling under this umbrella of our research question. For our proposals, what we will do, and this is actually what I normally do in the proposals that I actually write, is write two aims related to my research question. So we can have aim 1 and aim 2. Okay. Again, building on this same example that we've been working with, if our research idea is, quote, to detail microbiome characteristics related to warm El Niño conditions in juvenile red abalone Haliotis rufescens. Okay, so what I have in red here, in our research idea, what it says is microbiome characteristics. So really that part isn't so specific. So we talked in the previous video about how we specified the El Niño conditions and that they were juveniles, so that's fine. Those specifics are good, but other parts here for the microbiome characteristics, that part is a little bit broader, but that's fine because what we're gonna do is now in our specific aims, talk about more specifically what aspects of those microbiome characteristics we mean and how specifically we're going to address it. So what I'm going to show in the next slide is two aims that I wrote that specifically describe what is going to be done in the lab or in the field to address that broader research question that has been introduced. So this is one aim, the first aim of a proposal that I wrote. It says, quote, assess the gut microbiome before versus after heat stress in juvenile Haliotis rufescens. So again, we said generally in the research question that we wanted to look at microbiome characteristics. So specifically in this first aim, the additional information we're giving is that we want to assess the gut microbiome, so basically see who is there, what bacterial species are there, before the juvenile abalone have been exposed to warm temperatures, and then also after they've been exposed to warm temperatures. So this is a specific aim that is saying specifically what's going to be done to assess those microbiome characteristics. The second aim is then to determine if the composition and metabolic activity of the microbiome differs in heat-tolerant and heat-susceptible juveniles. And what this first part here in red, composition and metabolic activity, this is giving us more details about what kind of information we are going to get from the study we're doing. So this particular study that I am using as an example is a metagenomics study. And if you remember from our species module, we talked about one of the advantages of metagenomics is that it's able to tell you, first of all, what type of bacterial species are there, so who is there, but also what they're doing. So the metagenomics, because it's sequencing all of the genes that are there, it can also give you information about not only the barcodes, for instance, that can tell you what species are there, but also information about the functional genes that are contained within those bacteria, so we can get a better idea about what they're actually doing metabolically within the microbiome. So that's the first part of this aim that's more specific when compared to the research question. The second part that's more specific is now we're also introducing this idea of comparing juveniles that are heat tolerant which means they survive after they're exposed to warm temperatures and exposed to heat stress, and those that are heat susceptible, which means that they die after being exposed to heat stress. So in this second aim here, we're giving, again, specifics about what we're going to be doing in our study to address that broader research question. Here, those specifics are we're going to compare individuals that live versus individuals that die. And for the gut microbiome of each of those two categories of individuals, we're going to look at both what type of bacteria are there and what those bacteria are doing. And here, I want to take a step back and think more broadly about how your specific aims relate to your research question. And I think this analogy of the Goldilocks just right is relevant. So what I mean by that is I said we wanted our research question to have some specifics in it, but it also needs to be broad enough to encompass both of your specific aims. So it's important to think about here, we want our research question to be just right? So we need it to have some specifics, but we also need it to be broad enough so where two specific aims you write can be related to that slightly broader research question. So what do I mean by that? I'll show an example to illustrate what I'm talking about. So for this example, let's think about fish eggs. So as we've discussed in the species module, DNA barcoding is one way that you can identify fish eggs. So this is actually a study that has been done, by the way, Duke et al. 2018, I think, if you're curious and want to read this paper. So in this study, one potential way that you could formulate a research idea or a research question is to state the following, what's shown on this slide. So you could say something like, quote, "This research will use DNA barcoding to identify fish eggs and assess interannual variation in spawning in San Diego marine protected areas." So what I'm showing in red here are two areas where you can dive deeper in these specific aims to provide some specifics about how you're going to conduct this study and actually answer this broader research question. So for this particular study, if you focus first on this interannual variation part, what you can do is come up with an aim like so. So this is one specific aim related to this broader research question. This aim is to, quote, "document changes in spawning activity associated with changing ocean temperatures." So in the broader research idea, it introduces the fact that we're interested in this variation between different years. And in the aim, we're specifying even further that really what we're interested in is how temperatures may change across years. And unfortunately, we know that's definitely trending towards getting hotter. And so we can see then how those changing temperatures then might be associated with changes in spawning activity. So that's the first aim. The second aim then can delve deeper into the marine protected areas aspect. So what you can do is come up with a second specific aim, which would be something like this. Assess habitat-specific patterns of spawning in kelp forests versus sandy beaches. So here we're saying within this marine protected area, we're also interested in looking at how habitat differences within that marine protected area might affect different spawning patterns. And so this is actually the case in San Diego, so where I did my PhD at Scripps. The Scripps Pier is on a sandy beach habitat substrate, and just a couple miles away, but still within that marine protected area, is kelp forest habitat. So here, this second aim is giving us specifics that are based off of this broader research question, which states that we're interested in looking at spawning patterns in marine protected areas. So for this case, both of these aims relate to this broader research question. What I mean by this Goldilocks example, so in this case, now I've modified the research question and now it's too specific. So what I mean by that is this research question says, quote, "This research will use DNA barcoding to identify fish eggs and assess habitat-specific spawning patterns." So now, if this were our research question, this research question would match our second aim. So we're still talking about habitat-specific spawning patterns, and now in this second aim, we're saying specifically we're interested in kelp forests and sandy beaches. So the second aim is fine. The problem is that now our first aim doesn't match our general research question anymore, because our general research question specifically states we're looking at these habitat-specific spawning patterns, and that's all it says. Now, where this first aim is talking about changing temperatures, it doesn't make sense anymore because changing temperatures don't really relate to habitat-specific things. So this is what I mean by making sure that your research question contains some specifics, but is broad enough to still have both aims related to it. In this case, really only the second aim matches, and now the first one seems kind of out of place. So what we'll do now is do a little practice quiz on Canvas to help you build on what you did in the previous quiz and now write some specific aims for that. And then in the main assignment for this module, you will use a previously identified knowledge gap from a paper you found from one of the previous assignments we've done, and then formulate your own research idea and aims related to that. And so it's possible that will take a little bit of tweaking, which is totally fine. That's actually a component of that main assignment for this week, is you're going to post it on a discussion board, and then I will give you feedback and your peers will also provide you feedback. So again, this whole process of proposal writing requires constant feedback and changing and improvement. And so I don't expect your research idea and aims to be perfect right now, not at all. The idea is just to provide a starting point, and then we'll work from there and answer questions you might still have.